Hello, everyone, and welcome to today's ARC Talks webinar on the role of gene editing and hereditary TTR amyloidosis. I'm Isabel Lusada, and I'm the founder and CEO of the Amyloidosis Research Consortium, or ARC. Before we get started, I want to just point out a few features on this webinar platform. Firstly, we found that attendees using Google Chrome or Firefox tend to have the best experience. So if you're using another browser, you might experience some audio or visual issues. If that happens, try refreshing your browser or switching to a more compatible one like Google Chrome. If you have any questions, please type them in the Q&A box at the bottom right-hand corner of your screen, and Laurie from ARC will help address any technical issues. Any other questions we'll have for today's presentations will be addressed in the last 15 to 20 minutes of this hour. You can resize or hide any of the content boxes to suit your preferences. There is a box with resources that will have some relevant material to today's webinar. So just a little background on ARC. The Amyloidosis Research Consortium, ARC, was founded in 2015 with a mission to accelerate the development of and access to new and innovative treatments for all types of systemic amyloidosis and improve quality of life for patients. We work very closely with patients, patient organizations, pharmaceutical companies, regulatory agencies, and the researchers and clinicians who drive our mission forwards. Our four main areas of focus are improving speed and accuracy of diagnosis, increasing our understanding of the science of the diseases, accelerating regulatory approval for effective treatments, and enhancing care and quality of life for patients. We're so excited for today's webinar. The role of gene editing in hATTR presents a whole new frontier and real promise. This has been a really hot topic lately, and I'm sure many of you have heard or seen information about the ongoing trial from Intellia Therapeutics on the news and other sources, involving a type of gene editing referred to as CRISPR. And early results from the trial were shared this summer and drew major attention. So we organized this webinar to ensure the community has all the information they need and provide an opportunity to ask questions. We're really honored to have Professor Julian Gillmore presenting on this topic today. Professor Gillmore is the head and research lead at the National Amyloidosis Centre in London, also known as the NAC, which is one of the world's leading diagnostic and treatment centers for amyloidosis. Professor Gillmore is also the national coordinating investigator for Intellia's phase 1 clinical trial. And after Professor Gillmore's presentation, we'll take time to address questions. And joining us will be Dr. Liron Walsh from Intellia on the panel. So he'll be able to answer particularly questions that around trial and timing and things like that. Dr. Walsh is a nephrologist by training and is now the head of development for In Vivo programs. Thank you both for being here. Professor Gillmore, begin whenever you're ready.
Thank you very much, Isabel, and thank you to ARC for inviting me to speak. It's a great honor, and welcome to all of you. So I will present very early data from the first human trial of a CRISPR-Cas9-based gene editing therapy to be administered by intravenous infusion. The drug itself is known as NTLA-2001, and it's designed to edit the TTR gene in patients with transthyretin amyloidosis. So I'm going to start by asking you a question, actually, just to perhaps gauge where you all are, as it were. So how would you rate your current level of understanding of gene editing? I'll leave this up for a little while, and please press the relevant button, as it were, and then we'll see the results in a moment. numbers are continuing to rise, so I'll leave it a bit longer. Okay, I'll move to the results, if that's okay. Please vote if you haven't already. Okay, so about half of you said I know a little bit, and then just under a quarter each sort of I feel pretty knowledgeable, or this is brand new to me. That's and a few people with complete understanding, which is very impressive indeed. So, as you all know, hereditary transthyretin amyloidosis is a rare progressive disease caused by amyloid deposits which are composed of misfolded transthyretin or TTR protein, particularly within the heart and nerves. The main therapeutic strategy, which is successful in all types of amyloidosis that are amenable to treatment, is to reduce the concentration of the circulating amyloid-forming protein, in this case transthyretin or TTR. Existing gene silencing therapies reduce serum TTR concentration by approximately 80%, and they've been shown to stabilize and in some patients even improve neuropathy scores in hereditary ATTR amyloidosis. However, they do require chronic and lifelong administration. And on the basis of every type of amyloidosis that has ever been treated so far, one would expect greater TTR knockdown to facilitate clinically meaningful symptomatic improvements accompanied by better outcomes. So editing of the TTR gene, which has the potential to provide permanent and profound TTR knockdown after a single infusion, is a particularly attractive treatment strategy. This slide demonstrates the importance of the magnitude of TTR or magnitude of knockdown of the relevant fibril precursor protein. And I think it's a really important slide in relation to what I'm going to talk to you about today. So in systemic AL amyloidosis on the left here, you can see that outcomes are best in patients who achieve what we call, or what hematologists call, a CR, or a complete response. And that's equivalent, if you like, to 100% knockdown of the relevant amyloid protein, which in the case of AL amyloidosis is a light chain. And patients who achieve 100% knockdown do better than those who achieve what we call a VGPR, a very good partial response, which is equivalent to about a 90% knockdown of the relevant protein. And in turn, they do better than patients who achieve a partial response, which is a 50% knockdown of the relevant protein, who in turn do better than those who don't achieve any response at all. In AA amyloidosis, so a different type of amyloidosis in which the amyloid protein is known as serum amyloid A protein, this shows the relative risk, and I apologize for showing you these mortality slides. It's never very pleasant to show you these, but it's really to make an important point, and that is that the relative risk of death in relation to the octiles of serum amyloid A protein concentration in this condition, AA amyloidosis. And you can see here that the relative risk of death is nearly 4 times higher in patients who have an SAA concentration of between 4 and 9 milligrams per liter, as opposed to less than 4. Less than 4 is normal. 4 to 9 is very mildly abnormal. So you might, again, look at that as a 100% response compared to a 90% response. And you can see, as the response, if you like, diminishes, the risk of mortality increases. And here in ATTR amyloidosis, similar data is emerging, although it is very early days in terms of the history of treatment of ATTR amyloidosis. But this slide really makes the point that the more you knock down the relevant protein, the better the outcome, which is very relevant to our discussion today. So NTLA-2001 is a CRISPR-Cas9 based in vivo gene editing therapy. In other words, it's a gene editing therapy that can be administered by intravenous infusion. It consists of a single guide RNA molecule that targets the TTR gene. So you could think of that, if you like, as the homing device, and an mRNA sequence of strep pyogenes Cas9 protein. And you can think of that, or what that results in, the production of the Cas9 protein, as the scissors, okay? And I'll explain those two terms. So the homing device and the scissors, encapsulated in what we call a lipid nanoparticle, or LNP. So following intravenous infusion of this LNP, or lipid nanoparticle, it's transported directly to the liver, where it's taken up. Once it gets inside the liver cell, the Cas9 mRNA is what we call translated. It is translated to produce the actual Cas9 enzyme, the Cas9 protein, if you like. And that interacts with the single guide RNA to form what we call a CRISPR-Cas9 ribonuclear protein complex. It's a mouthful, but that's the name of the complex. And the RNP complex enters the nucleus of the cell, and the guide RNA binds to the DNA double helix at the target site. In other words, it binds very specifically to the TTR gene in the nucleus. And that leads to precise cleavage, or a cut in the gene, and consequently that leads to a reduction in production of the TTR protein. I hope that's clear. So the plot here shows you the effects of concentration of the guide RNA on the percentage TTR gene editing by NTLA-2001 in liver cells in the laboratory, what we call cultured primary human hepatocytes. So these are liver cells in the laboratory. This vertical line here marks the concentration at which we routinely observe 90% reduction in TTR protein production, known as the EC90. And above the EC90, what you see here is that NTLA-2001 achieved saturating levels of TTR gene editing. And these were accompanied by greater than 95% reduction in TTR protein production, if you like. So this gene editing resulted in a marked reduction of TTR protein production from these cultured human liver cells. Now, an important question that the scientific team asked before being able to think about administering this drug to patients was, what is the level of what we call off-target editing? In other words, editing of genes that you don't want to edit. You know, what you want here with this drug is to very specifically edit the TTR gene, the target gene, but you don't want to edit other genes that are in the nucleus. So what's the level of target editing at these concentrations? And I won't go into lots of detail, but the team at Intellia performed extensive whole genome screening using a variety of techniques, a combination of computational techniques, laboratory-based techniques with and without cells, basically to detect and validate the likeliest sites for potential off-target editing of a gene that one doesn't really want to edit. And these methods identified 7 potential sites, but they were all in what we call non-coding regions of the genome. In other words, regions of the genome that are redundant in terms of protein production. And what this experiment is designed to show is to compare the rate of on-target, in other words, editing of the TTR gene, and off-target editing with NTLA-2001. What you see marked down here by these colored lines, each representing one of those 7 sites that was identified, is that there was no detectable off-target editing, even at concentrations of NTLA-2001 3 times greater than the EC90. So extraordinarily reassuring data in terms of a risk of editing genes that one wouldn't want to edit. So then preclinical in vivo in the body, if you like, studies were conducted in a monkey species, cynomolgus monkeys. And this graph shows the mean percentage reduction from baseline in serum TTR protein concentration in monkeys who received the lipid nanoparticle at 0, 1.5, 3, and 6 milligrams per kilogram of the drug. And they received the drug on day 0, and they were followed for 367 days. Note, a single dose of 3 milligrams per kilogram achieved a durable reduction in circulating TTR protein of approximately 95% after day 28. Safety studies established a dose range at which no adverse effects of NTLA-2001 were observed, with near complete sustained reduction in serum TTR following a single administration. At higher doses, adverse effects in the monkeys were dose-dependent, transient, reversible, and monitorable. So the first in-human study with NTLA-2001 is a 2-part, open-label, multi-center study in patients with hereditary ATTR amyloidosis and polyneuropathy. Part 1 is what we call a single ascending dose design with a minimum of 3 patients in each of 4 dose escalation cohorts. The recommended dose from Part 1 will be administered in a dose, what we call a dose expansion cohort in Part 2. And the primary objectives of this first in-human study are to evaluate safety, tolerability, and pharmacokinetics and pharmacodynamics, the latter by measurement of serum TTR levels. So I'll report data on the first 6 patients from the first of 2 dose escalation cohorts included in Part 1. Bear in mind the participants received a one-time infusion of NTLA-2001 on day 1 of the study. So here are the demographics of these first 6 patients, 3 in each of the 2 dose cohorts. The age range from 46 to 64. 3 different pathogenic TTR mutations were represented, and 3 patients received prior diflunisal therapy. All had early stage polyneuropathy, and despite the presence of cardiac ATTR amyloid by imaging in 5 of the 6 patients, accompanied in some by elevated NT-proBNP, the cardiac biomarker in the bloodstream, they were all NYHA or functional class 1. In other words, they essentially all had early stage disease. The median time from diagnosis to treatment in these 6 patients was 2.5 years. So NTLA-2001 was generally well tolerated in the acute phase, in other words, in terms of the time period that these patients were followed, with adverse effects all being what we call grade 1, so low grade, and mostly unrelated to treatment. One patient did have a mild infusion-related reaction, but it didn't require dose interruption. And importantly, there were no serious adverse effects. There was no evidence of liver toxicity. These are liver blood tests and coagulation blood tests. And there was absolutely no evidence of liver toxicity or problems in terms of blood clotting, which can be a complication of liver dysfunction. And all laboratory abnormalities were clinically insignificant. So serum TTR reduction was observed by day 7 and deepened further by day 28 in all patients at the first dose level of 0.1 milligram per kilogram. And the reductions ranged in this cohort from 47 to 56%. The reductions were dose-dependent, with greater reductions detected in the 3 patients who received the higher 0.3 milligram per kilogram infusions. Here, the reductions range from 80 to 96%. At day 28, mean TTR reduction from baseline in the 0.1 milligram per kilogram cohort was 52%, and in the 0.3 milligram per kilogram cohort was 87%. So, in conclusion, systemic administration of NTLA-2001 in patients with hereditary ATTR amyloidosis and polyneuropathy caused a profound and dose-dependent reduction in serum TTR protein concentration. Of note, maximum reductions were 56% and 96% respectively in the 2-dose cohorts. NTLA-2001 treatment was well tolerated in the acute phase, with all adverse effects being of mild severity. Further dose escalation in this ongoing first-in-human study is planned with the aim of achieving greater reduction in TTR than that provided by currently available agents. And that's expected to translate into improved clinical benefit for patients, as I mentioned in my first slide. In other words, improvement in autonomic symptoms, better mobility, fewer cardiac hospitalizations for heart failure. It's noteworthy, I think, that the prognosis in ATTR amyloidosis is very much driven by the cardiomyopathy, by the heart disease. And it's certainly conceivable, I think, that the greatest benefit of profound TTR knockdown, as we're seeing here, will be in patients with cardiomyopathy. But that at this stage is pure speculation. So this is the first demonstration of CRISPR-based in vivo gene editing in humans, and these findings provide proof of concept for a promising new therapeutic strategy. So it just leaves me to thank or acknowledge the patients and their families, the contract research organizations involved in the trial, my co-investigators at the National Amyloidosis Centre, Richmond Pharmacology, and at the University of Auckland, and of course, most importantly, perhaps the clever people, the many members of Intellia Therapeutics and Regeneron Pharmaceuticals who were responsible for the development of NTLA-2001. So thank you very much for listening, and please don't hesitate to ask me any questions.
Thank you, Professor Gillmore. That was really excellent presentation and of a complex concept, and the results are certainly very exciting, and we look forward to further development. And I'd love to ask Dr. Walsh to join us for the questions that we have quite a lot and more coming in. And Laurie, can we lose the slides and then we can go to full screen? And I will start off with a question which is an interesting one, that while it's currently being investigated in hereditary ATTR, what about wild type?
Yeah, so obviously a very important question, and we do know that wild type ATTR amyloidosis is more common than hereditary ATTR amyloidosis. So absolutely, the plan is to, I suppose the first thing to say is that in principle, one would expect this treatment to be effective in wild type ATTR amyloidosis because reducing the concentration of the TTR protein ought to be effective for both wild type and hereditary ATTR amyloidosis. And as I alluded to at the end, it's very possible that, in fact, the benefit will be greatest in patients with predominant cardiomyopathy, of which, the patients with wild type ATTR amyloidosis obviously constitute a major cohort. So the plan is to test the drug in wild type as well as predominantly cardiomyopathic hereditary ATTR amyloidosis. For example, the mutation, the V122I mutation, or variant, which is present in 4% of people of African descent or Afro-Caribbean descent.
And following on from that question, we've also had a question about other types of amyloidosis, such as AL or some of the other hereditary but non-TTR types.
Yeah, I mean, there's certainly, so I suppose the first thing to say is that, there are obviously multiple potential applications of CRISPR-Cas9 gene editing. It's a modular platform. One can technically aim to edit any gene in the body. And the challenge really is being able to deliver that treatment to the relevant cells, which express that gene. In other words, the relevant cells in which that gene produces a protein, if you like. In the case of ATTR amyloidosis, TTR is exclusively, or circulating TTR, I should say, is exclusively produced in the liver. And that is one of the reasons that this was a good target as a first treatment of this technology, because the lipid nanoparticle enabled us to develop, or the company, I should say, to develop a drug that gets into the liver very effectively, and that's what this trial has shown. In other types of amyloid, it's early days. There are, excuse me, there are theoretical possibilities in at least one other type of hereditary amyloidosis, and there are theoretical possibilities in terms of AL amyloidosis, but none of those at the moment are either in development, as far as I'm aware, or likely, I would say, to be in development anytime soon.
Thanks, Doctor. Can you hear me? It's Liron.
Yes.
Yeah, great. Thanks, Dr. Gillmore. Yeah, that's perfect. I don't have much to add other than to say that, this is Intellia's first approach into the gene editing for a serious and debilitating disease. And we do hope that we can expand in the future beyond what's sort of presented today, and hopefully that will come. I know that the patients are waiting for new options beyond amyloidosis even. So this is something that we're keen to bring forward.
And this is an excellent next question. We've had. Oh, go ahead, Dr. Gillmore.
If I can just perhaps just temper expectations. I think it's important for us to do that. And I think, obviously this is extraordinarily exciting data, but, I do have to say to all of you that this is very, very preliminary data. It's 6 patients and it's early stage data. And, obviously it's extraordinarily exciting, but at the same time, as we all know, and as you all know better than I do, and as Isabel alluded to right at the start, one of the aims of ARC is to speed that whole process of, getting new drugs to patients, which can take time. So I think that, we do need to acknowledge that obviously the trials need to be run, and that is going to take time, and development of any new drug is going to take time. So whilst it's amazingly exciting, I think for all of us, we just need to temper it with those expectations.
And it might be very helpful to hear a little bit both about the process, so the different phases of trials, and when we're looking to develop a product for approval, and how Intellia is thinking about approaching that. We've had questions about whether studies will be open in the US, even in Scotland. And so just a little about that, and how, very theoretically, but, what is the time expectation of a standard trial design, and how long that might take?
Liron, do you want to take that? Or? Yeah.
Sure, yeah. Of course, Dr. Gillmore. So I think it's a great question and a really important one. And I'll piggyback off really what Dr. Gillmore mentioned in his sort of last remarks related to this is early data and there is time that's needed to bring forward this therapy in a responsible manner. Right now, we're in a phase 1 study, and we are looking to complete that study in the not too distant future, and then thinking about further phases of development. And in those further phases, we hope that we'll be able to bring to test the drug in the United States and other regions and countries as well. With respect to the sort of the process here, so as we think about it, we're thinking about a phase 1 study to understand the preliminary safety as well as how and to what extent we can knock down TTR, the critical agent that is causing disease in these patients. When we've had a chance to review that safety and that efficacy data, we'll be able to bring that to health authorities and talk about further development. We hope that we can bring that forward again in the not too distant future, but we want to make sure that given the new advances that Intellia is bringing forward, that we understand the potential for benefit as well as whatever risks there may be. I'm hoping that with the advocacy of patient organizations, that we can bring forward therapies in a manner that we can impact patients' lives in a really dramatic fashion.
Thank you. There's a question that said, if you participate in a trial and received a low dose of the CRISPR, can you later get onto the right dosage, or could you continue on another drug? What's the plan for those patients?
Yeah, I mean, the Intellia have been very, very clear from the start that they intend to give the patients who've received, if you like, a suboptimal dose in terms of the ascending dose, the full dose once they know that it's safe to do so. So in this situation, in a first-in-human study, which is a point at which you're trying to establish, one of the purposes of that first-in-human study is to establish the optimal dose. And obviously, as you could see from the TTR knockdown in the first group of patients, they have the potential to achieve higher knockdown, which we believe to be associated with better outcomes, as I outlined. And Intellia, as I say, have been very clear that those patients will get the optimal dose once it's been selected, as long as it's safe to do so.
And if there's a patient who is currently on tafamidis and mentions about the knocking down the thyroxine, and is that the same case with this? And are there any thoughts about how that can be replaced in the body?
So in terms of, I suppose your question, if I understand it rightly, refers to the physiological role of transthyretin and whether knocking it down could cause some harm. And I think the important thing to say there, so what we know about TTR is that it transports thyroxine and vitamin A. And there is quite a lot of overlap in those functions. But nonetheless, all patients on patisiran and inotersen, on the gene silencers, which work in the same fashion or which also knock down TTR, if you like, they are on vitamin A replacement, as are the patients in this first-in-human study. So by definition, the patients need vitamin A replacement. I think that, what we can say is that we've got experience now of, nearly 10 years of some patients who have had a roughly 90% knockdown. Some patients on the gene silencers achieve somewhere in the region of 90% knockdown. That's not the mean or the average, as I pointed out earlier, but a few do. And the ones who have achieved that knockdown, we really don't have any suggestion of toxicity related to low TTR levels. So that's obviously reassuring. Now, we don't know, the honest truth is that we don't know if we achieve 96% knockdown, will that cause harm? We don't think so. And, I think the data with the gene silencing drugs is very reassuring. But at the same time, that is another thing that will need to be followed in terms of the longer term.
Thanks. And there's a question which, a couple of questions around the ethics of gene editing. And I think we've heard over the years a lot in the news about this. And I think just understanding how this works, will it impact the next generation? How long does it last? Things like that be helpful to have addressed.
Yeah, it's a very important question, isn't it? I mean, I think obviously, the aim with these treatments is simply to better, to treat disease and edit genes that cause human disease. And I think one really important thing is that there is, the company Intellia have to abide and do abide by, the internationally developed principles for the ethical use of genome editing technology. And what that means is that editing is strictly limited to what we call somatic editing and not germline editing. So to explain that term, you cannot edit a gene, if you like, that gets into cells that could be transferred to an offspring. So so essentially, you're limited to editing genes in that individual patient that won't affect their offspring directly. So I hope that. I hope that explains. Obviously, there's a lot more. One could talk all day about the ethics of this technology, but I think the important message is really as that editing is somatic editing, and obviously there are important rules which the company have to abide by to do with the ethics of the technology. I don't know if Liron may want to add anything there.
Yeah, I think your summary was great, Dr. Gillmore. I think it comes back to responsible people in collaboration with investigators such as yourself and health authorities to make sure that we are moving forward in a manner which ensures that patients are fully informed and that things are safe. And so we are following sort of the standards that are set by both the FDA and Europeans in terms of ensuring that patients are protected.
Thank you. And along that line, there's some questions around, first of all, how long this will last. And also the question of if at some point in the future it was discovered that something critical had been deleted, is there a way that the process could be reversed?
So in terms of how long it will last, the data that's available, I showed you from the non-human primate study. And that showed, persistent knockdown after 367 days, as you saw with a single infusion. And the other evidence that this may indeed be permanent, so we suspect that this may be permanent. And the other evidence for that is that when a surrogate NTLA-2001 or a surrogate LMP was given to mice, their livers, 2/3 of their livers were removed. After regeneration of those mouse livers, the TTR knockdown persisted. So what that's telling you is that even after regeneration of the liver, there's persistent TTR knockdown. So that's kind of good evidence that this may well indeed be permanent. But again, that is data that we don't have in humans yet, and that will need to be seen. To answer the second question, two things. One is potentially the drug could be administered again, as far as we're aware. although more studies do need to be performed by Intellia in terms of re-administration, but there's no reason to suspect that that wouldn't be possible. And then the third point is that if you discovered, for example, that knocking down TTR by 98% was a bad thing to do, you would have the potential, using the gene editing technology, not exactly the same technology, but using the gene editing technology, you would have the potential to reintroduce, for example, the wild type, the normal TTR gene.
And for patients that have CNS or ocular involvement, is this going to impact them, or will it help?
So that's a really important question, and this particular treatment is not going to help them because this is aimed at liver-derived transthyretin, and amyloid in the vitreous and leptomeningeal amyloid deposits are produced in the eye and in the choroid plexus. The TTR is actually produced in the eye and the choroid plexus, and this particular drug doesn't get that. So NTLA-2001 wouldn't help those particular patients.
Mm-hmm. We've had quite a few questions about the trial, phase 2/3 trial design. I don't know if you have any information you can share. Certainly patients are interested if they have cardiac involvement or have had a heart transplant, if they've been on a prior or are currently on a treatment.
So I can, sure, maybe I'll take that, Dr. Gillmore. As we've said, we're still in sort of the early stages of understanding the therapy within the context of a phase 1 study. We do hope to move into patients with cardiomyopathy in the future, and the design of those studies are still under consideration. And so we'll be, back in the future with some more details with respect to that. But that is the aim, really, is to provide the therapy to polyneuropathy and patients with cardiac involvement in the future. And I know patients are waiting, and as soon as we can make that happen, again, responsively, we will certainly do so.
And do you see a trial design, and is it important to consider testing this against the current treatment landscape, current treatments?
Yeah, that's another great question. I think that'll be part of the considerations as we move towards a pivotal trial design. I think there are complicated issues with respect to that, but most certainly that will be amongst those that we consider. And
here's one saying, I'm on a messenger RNA manipulation medication. I've received my mRNA vaccine. Oh, no, I lost my question here. I just jumped around. And it looks like the future of disease treatment will involve our genomes in one way or another. Do you see a limit to the use of gene manipulating medications?
I think this, this has the potential to, change medicine very dramatically. That's not to say there aren't a huge number of challenges and a huge number of, developments that need to occur before one can use, gene manipulation or gene editing in a wide range of diseases. But there are already, a number of programs in various other conditions. And, in terms of, as I say, the real challenge is going to be delivering the technology, which after all is modular. As I said earlier, technically speaking, the drug itself can be targeted to any gene in any gene in the body, and you can manipulate any gene in the body. But the challenge is to get to the right cell, to deliver your drug to the right cell, so that it so that it stops the production or alters the production or changes the production of the relevant protein.
Excellent. And there was a question asking, is transthyretin protein also called prealbumin? That was mentioned.
Yeah, that's that's its old name. Yeah, indeed.
Excellent. Let me just see what else we had. And then there's a couple of questions around, do you want to have, with the level of knockdown of TTR, is there a sense that some percent should be left? Is total knockdown what we're looking for? What's a healthy amount?
I think that's it's obviously a really important question, isn't it? I mean, as I pointed out earlier, all the data in amyloidosis generally in other, based on other types of amyloidosis, is that the more you knock down the protein, the better the outcome. And that makes complete, if you like, common sense. Essentially what we're doing, if you've got no TTR around, let's say, in your bloodstream, you've got nothing to make more amyloid, full stop. And what we know from an imaging technique that we've been using here at the Amyloidosis Centre for quite a number of years is that the body does get rid of amyloid. It just does so very slowly. So in a patient who's producing it, they're usually producing it more quickly than they're getting rid of amyloid, okay? But if you knock down TTR by 100% in the bloodstream and you're not making any amyloid, by definition, you'll be getting rid of it more quickly than you're making it. And that's the principle of treatment. And so, we believe that knocking down by, 100% from an amyloidosis perspective would be excellent. But what we really don't know is, do you need to have some TTR around for some crucial physiological function that we're unaware of? So I think, as I said earlier, I think what we can be reassured by is those patients who have achieved 90 to 95% reduction on gene silencing treatments so far. We really haven't seen any complications associated with those reductions. But we need to, it's relatively early days. That's gene silencing drugs have only really been, or patients have only been on them for maybe, 6 to 10 years. So, we need to follow these patients up for longer. And I think everything's encouraging at the moment, but let's wait and see.
So I think this is a good follow-on. There's a patient here who is on both tafamidis and patisiran and has low vitamin A and has tried a number of replacements with minimal success. Is there a form of replacement therapy that gives optimal results?
So I think one of the things to say is that if a patient is on, the first thing to say is that the assay for vitamin A measures vitamin A that's bound to, for example, transthyretin, as well as vitamin A that is free. And with most proteins, it's the free bit of the protein that performs the physiological function. So by definition, if you reduce TTR by 80%, when you measure, vitamin A, you're measuring the bound component, which is low because the TTR level is low. But that doesn't necessarily mean that the free vitamin A is actually low. So, and it's the free vitamin A that is probably performing the physiological function. So I think that, it's one thing to measure vitamin A levels, but at the moment, we have as I keep mentioning, we have no indication that patients who have low vitamin A levels, who are on vitamin A replacement, have any problems in relation to their eyes after many years of patisiran. So I think the vitamin A level as such is it's a surrogate. I'm not saying it shouldn't be measured, but I think the more important thing is having a low vitamin A level harmful in this context? And we've not got no indication that it is.
Thank you. Does the lipid nanoparticle eventually leave the body? And somebody had asked this question saying they've heard that it can cause reactions.
Yeah, so the lipid nanoparticle does get broken down, if you like, and leaves the body. It can, lipid nanoparticles can certainly be associated with reactions. And as I say, what we can say on the basis of the data I showed you is that, so far the doses that have been administered have been extremely well tolerated. But again, that's something that, in the dose escalation study, we'll need to keep monitoring.
So here is a more scientific question. For the guide RNAs that were designed, is it better to design a single guide or two guides targeting the start and stop codons? Do they impact the off-target efficiency?
So go on, Liron. I'm sure you will answer that better than I was.
No, I'm not so sure about that. I think it's a great question. I'm not sure if this was sort of explored. What we know that based on the guide that was developed or the surrogate from that was used in the non-human primates is that we get really efficient editing and knockdown. And so rather than introduce a second agent, this is the methodology that we're exploring at the moment. Perhaps something to consider down the line, but at the moment, I think we're quite pleased with the results that we've achieved, and we hope to maintain this path forward.
I think one other thing that's worth adding to Liron's excellent explanation is that the company selected a part of the TTR gene that is not known to contain any pathogenic mutation, so that one would expect, one would expect to achieve TTR knockdown with all the known pathogenic mutations of TTR equally, if you like. So there's no suggestion that a patient with one particular mutation might get a better knockdown than a patient with a different mutation, because the guide RNA is targeting an area of the TTR gene that is not known to be mutated in any patient with the disease.
Which would also mean for those with wild type forms of amyloidosis as well. So thanks for adding that, Dr. Gillmore.
And there's some questions around what about the organ where there's already amyloid deposited? Will this remove the amyloid deposition? Or is there potentially a need for another treatment that might remove those deposits?
Yeah, so that's an excellent question. As I said earlier, all amyloid deposits in the body are in an equilibrium between the rate of production or rate of new amyloid formation and the rate of removal. And the rate of removal in the human body is by definition very slow. It does differ from one patient to the next. Some patients get rid of their amyloid, relatively speaking, far more quickly than other patients. But by definition, all patients get rid of amyloid slowly. And the other thing to mention is that the rate of removal from different organs does also differ. And, there is no doubt that, here, what we're doing with this particular treatment and indeed with the gene silencing treatments, as I alluded to earlier, is we are, trying to stop ongoing production. But there is no doubt that targeting the other side of the equilibrium, if you like, to speed amyloid removal would be an attractive additional treatment, or have benefit in terms of speeding removal of existing amyloid.
Mm-hmm. So here's an interesting question. Did the mice and monkeys used within the study normally develop hATTR amyloidosis, or were those animals altered to develop that condition?
No, so these were, the monkeys. So in fact, the LNP, if you like, was a slightly different LNP. It was altered. It was what we call a surrogate LNP that specifically targeted the TTR gene of the monkey. Do you see what I mean? So the monkeys potentially had a normal TTR gene sequence, didn't necessarily get amyloid, but the principle here was to show that you could deliver it to the liver and knock down TTR in the bloodstream. So it wasn't specifically related to amyloid.
And aside from the need for vitamin A therapy, there were two questions. One is, does diet in any manner affect either positively or negative the ultimate outcome? And also the follow to that was, we talked then about vitamin A being replaced by a supplement. How is thyroxine replaced? And does it need to be?
So I'll answer your second question first. I mean, thyroxine replacement, thyroid function is being monitored within the study. And, at the moment, the answer to your question is no. But again, that will be, a subject of ongoing monitoring. In terms of the first question, of course, diet is, very, very relevant in hereditary ATTR amyloidosis, particularly in patients who may have autonomic neuropathy, involvement of the autonomic nerves, and tend to lose weight. So although it's not necessarily directly linked to the question of this particular treatment, diet is obviously an extremely important component of the disease itself and outcome in relation to the disease.
Thank you. So now back to a slightly more scientific question. What directs the lipid nanoparticle to liver as opposed to other cells?
Liron, I don't know whether you're more qualified than me to answer that one. Yeah.
No, I doubt it. I'll give the sort of the simple explanation is that the lipid nanoparticle is able to be targeted towards the liver based on receptors that are on sort of the liver, and therefore the uptake of the lipid nanoparticle is predominantly within the liver. Similar in terms of sort of LDL cholesterol, and in terms of the honing in, so to speak, onto the liver.
Excellent. So we have had a huge amount of questions today. We've tried to answer the major themes. If there were some questions that were missed, we will be going through those afterwards, and we will send them to Professor Gillmore and Dr. Walsh to confirm and to answer, if that's all right with you. I'd like to take a moment to thank you enormously for today's presentation. It's really exciting. This is, to remind everyone, this is very, very early data, but it is very encouraging. I know that there have been a lot of questions about the clinical trial design and who will be eligible and who won't, whether patients could be on a placebo or whether there'll be another treatment they'll be on, what type of amyloidosis, how advanced you might be. These are all things that Intellia with the scientific community will be looking to address. We at ARC will keep patients informed of the trial design and information about the trial as it opens and becomes available in different locations. So if you have not done so, sign up to myamyloidosispathfinder.org and put your information in, and you'll be notified as the trials open and trials that might be relevant for you. Again, thank you so much for sharing this information, and we really, really enlightening, and I'm continually impressed by our patient community on the level of information, both the knowledge that they have and the interest. So thank you really for attending this webinar and for asking excellent questions. Thank you very much, Dr. Walsh and Professor Gillmore.
Thank you very much, Isabel.
Thank you.
And second my thanks.
Yeah, wonderful to participate, and very much appreciate the patient community's involvement in progressing new therapies. Big thank you.
Thank you.